Welcome you all to this Cryo-EM course, and today there is a practical session. As I mentioned during my class that we are going to show you the practical session where we will discuss about the TEM transmission electron microscope, cryo-electron microscope, as well as the sample preparation, how we are preparing the grids that we are going to show you. So this is a Cryo-EM facility at IISc Bangalore, where you can see this is a cryo-electron microscope that is called the Talos Artica. So this Talos Artica, it is from Thermo Fisher Scientific, and this Talos Artica is a 200 kV microscope. You guys already know about that, what different types of microscopes are possible. So previously I already discussed about 120 kV microscope, 200 kV microscope, 300 kV microscope. This is a 200 kV cryo microscope where we have the cryo facility. This is, you can see that whatever I discuss in the class, you cannot see all these component anymore because most of the things are under this white box. So we'll open this white box and I'll show you each and every component of this microscope. Here you can see whenever you are going to insert the sample, you just have to open this window and then you can insert this sample here. You don't have any access to this entire microscope because whoever is the expert, they can only handle this microscope. Here you can see a window also. In that particular window, you can see lots of values are written. So sometime you can see the pressure readout. Previously I already discussed about that whenever we have a electron microscope, we need a very high vacuum, and that high vacuum you can monitor here, that here it is, you can see that accelerating stack or column or detection unit, autoloader, there you will get different kind of values and everything it will show you is under high vacuum. So those things it will be clear when you will look at the microscope interface, when you will start our imaging. That time we'll discuss those things more, but here during the sample insertion you can monitor those values also. Why it is important? Because I already discussed these things with you guys, that whenever we are inserting our sample or inserting our holder, that is the only point where you are breaking the vacuum to insert your grid. So you always have to monitor your vacuum when you are inserting your sample. As well as for the cryo electron microscope, temperature is an important part. If you break the vacuum, temperature will increase. If temperature will increase, that is going to affect your grid. You can get a different kind of ice contamination. If you follow that class, that why we are getting different kind of ice contamination, it will be clear to you guys, but same time you have to pay attention when you are inserting your sample. Now if you look at here also, that is also showing you the nitrogen level, that you have the column here and the autoloader that is maintaining certain amount of nitrogen level in that column and the autoloader. And here also you can see autoloader, vacuum and cassette, at what condition it is a docked condition and undocked condition. So some preliminary parameters you can see it in this window, and all the details you can see it where data collection center, where we are going to collect our data. There you can see all these details that we'll discuss in the later. First we'll talk about our microscope component, and then we'll discuss about the sample preparation, then we'll discuss about how to make the cartridge and insert inside the microscope, and then we'll talk about the data collection and how we can collect our data.
Good.
So now I am going to open this entire system, and I am going to show you individual components. As I mentioned earlier that okay, this entire system is within this white box and you cannot see anything from here, right? So the best way I'll open this entire system and then I'll show you what we have inside, because you already know whenever we are talking about the electron microscope, what we have electron gun, we have high tension, and then accelerating stack, then we have a condenser lens, objective lens, intermediate lens, and finally we are collecting the data using a direct electron detector. So now here in this white box you cannot see anything. That is the reason we have to open this box and we want to see it. But most of the facility you cannot see inside here. That is the reason I want to show you all the things in details in this class. Okay, so I'm opening and I'm showing you what will get it. First I'll open this one. Then there is a lock here. We'll open this entire part. Okay, so you can see the inside of that microscope. So now you can see it is a very complicated instrument. It is not that, a white box anymore. So if you look at this microscope, what you can see, this top part, the filament or gun, it will stay here, and accelerating stack will also stay here. Now if you come, this is the entire column that we are constantly talking about, that column part. This is the entire column. Now if you look at here, you have couple of parts here. So now you can see this is the entire column, and if you pay attention here, you can see a box here. The one box it is coming here, second box it is coming here, and third box it is coming here. In this box, basically those are the lens system where aperture is going in, and if you want to control everything manually, you control from those system. So one will be the condenser. So condenser lens system will be here, then your objective lens, and then your intermediate lens system is here. Now this particular setup is connected with a special setup here that is called the goniometer. Previously we already discussed about that one. That goniometer is a system where you can insert your sample. This is the goniometer. You can see it is a very complicated setup. So generally you cannot see goniometer like that. So in this particular case, this is the area where you can insert your sample, and there is a lot of parts are there which is going to control your holder tip. But in this particular microscope, this is the part it is blocked, because our system has an autoloader system which is connected at the opposite side of this goniometer. I'll show you when I'll go to that side. Now near this goniometer you can see there is a white tank is attached here. So in this white tank, it is basically a dewar. Those are the liquid nitrogen pipe which is connected with this dewar, and liquid nitrogen will fill here. All the time it is maintaining this liquid nitrogen. When I was showing in that window, there I told you that it is maintaining some nitrogen level, that okay 40% or 70%. That nitrogen level basically talking about this dewar, how much this dewar is filled. So this dewar will be filled by liquid nitrogen in every 8 hours and 3 times a day. So we should have a big liquid nitrogen dewar which will be automatically fill this dewar to maintain the temperature here. Why do we have to maintain the temperature here? Because we are going to insert sample in this particular region using that goniometer, right? When you will insert our sample there, we have to maintain that temperature properly, and that is the reason this liquid nitrogen dewar will be connected with the cryo box which will maintain the temperature. I already told you this is the goniometer, and now we are looking at this goniometer from the side. So I told you this goniometer, the sample insertion area, that hole is blocked, and that if you just go completely opposite 180 degree, you will find out this is the autoloader system, and you can see this one is connected with the column and sample is going through this. So in this recent microscope, most of the goniometer is blocked, and then they are connected with a autoloader. This L-shape instrument, it is called the autoloader. If you just look at this L shape, and you can see this window from there, you can see how grids are going in, and so here is the area where we will put that nano cap and grid will go inside through this one. So this we will discuss more when we will insert our sample or cassette. You can see how grid or cassette is going up and staying inside this autoloader. This entire system, this L-shaped entire system is the autoloader. And now you can see another dewar. The same dewar is attached with the autoloader, which is maintaining the temperature of the grid inside that autoloader. Now in this microscope, multiple pumps are attached. You can see IGP pump is attached here. This is one of the IGP pump, which is IGP pump means iron pump, that we already discussed about that, and it is called the trapping pump also. So this iron pump is basically maintaining the fine vacuum inside that column. Not only that, there is a turbomolecular pump is there. Turbomolecular pump, it will start from atmospheric pressure to very high vacuum, and then iron pump will start working. And you can see there are multiple other vacuums and different types of electronics is attached. So whenever we will touch this microscope, we have to be very careful. We cannot touch whatever component we want. We have to be very careful when you will handle this instrument, because if there is any problem with any electronics, then it will be very difficult to figure it out where is the problem, and this entire setup will be stop functioning. So it is better to hand over those things to the expert who can handle this entire system. So you can see a cable is coming here, very thick cable, and it is connected here. This is the high tension cable. It is coming from the high tension tank, and when it is connected here, it is going to the electron gun. Electron gun is staying here. We are looking at the same microscope from the back side now. So that is the reason this part is very much clear, where you can see the high tension cable is connected with the gun, and the after gun we have a accelerating stack and rest of the part. From here you cannot see autoloader anymore. You can only see that dewar which is connected near the goniometer. Now if you go down, you can see that different pumps are connected with this microscope. One important pump we are constantly talking about, that is called the turbomolecular pump. This is our turbomolecular pump which is connected with the microscope. When this turbomolecular pump will work, this is the turbomolecular pump. When this turbomolecular pump work, it will reach to a very high vacuum. Then iron pump, it will start. Now at the bottom, if you look, this is our K2 direct electron detector. This K2 direct electron detector, there is a CMOS camera that is called CETA camera, and the bottom there is a K2 camera is attached. Here we are using this K2 camera to collect the data. So now you can see that is the column. Goniometer, I already discussed about this. Now if you come all the way down, you can see something is attached here, right? So this is our CMOS camera, that is from the Thermo Fisher CMOS camera. It is called the CETA camera. It will attach with this microscope, and then at the bottom there is a K2 direct electron detector. That is the main direct electron detector we are using. So you can see this K2 and the K2 mountain symbol also. Based on that, it is called the K2 direct electron detector. And the another device is attached with this K2 camera, which one is the digitizer. So whatever data we are collecting, it is processing there, and then we are recording all the data. So here you can see this is the high tension tank. So generally for any kind of microscope you have a 200 kV or 300 kV or 120 kV, and you cannot take it from the normal power sources that 200 kilovolt or 300 kilovolt. So this is the tank where we are generating this 200 kilovolt, which is supplied by through this cable that I already showed you inside the microscope, how it is attached with that electron gun and the accelerating stack which is regulating here. And this is the main, this is a very important part where it is generating 200 kilovolt. If you don't have this unit, it will be very difficult to get that 200 kV microscope or 300 kV microscope or 120 kV microscope. Okay, so now you can see there is a liquid nitrogen dewar. This is a very big dewar, almost 250 liter. You can, if required, you can keep a bigger one also, but we have this kind of 3 to 50 liter dewar which is connected with the microscope. Here you can see one pipe, it is connected, and there is one valve, and there will be another valve here. So one line you will get it for gas nitrogen, and one you will get it, this one you will get it for liquid nitrogen. So this liquid nitrogen, if I just come here and I'll show you that this is basically for liquid nitrogen and this is for the gas. And liquid nitrogen will go and stay in this liquid nitrogen dewar that I already mentioned for the autoloader and the main dewar which is connected with the column. And what is the function of this gas? Generally, when you want to open any valve and want to load nano cap, or we want to insert the cassette or grid inside the microscope, that time this gas nitrogen is required. And here you can see this is the liquid nitrogen tube, it is going inside the microscope, and if there is a excess liquid nitrogen or gas, that will come out through this pipe. Okay, I already showed you the entire electron microscope, or you can say that cryo-electron microscope. Now I am going to show you the sample preparation unit. So this is our sample preparation unit. Sample preparation unit, you can say it in a two different ways. That one, where you are working in the laboratory, where you are doing your biochemistry, you are purifying your protein, that is a different types of sample preparation. Now, when you get your sample, means biological macromolecules, it may be bacteria, it may be cell, it may be your protein, maybe DNA protein complex or RNA protein complex, you are getting that, and now you are going to add those samples in your grid, and you are going to prepare a cryo-EM grid. That is called the sample preparation for cryo-EM, and that sample preparation we are going to do using this instrument. This instrument, it is called VitroBot. This is a Mark IV VitroBot unit, where you can see there are different parts. It is a semi-automated system where you need some kind of manual part, that where you are going to insert your tweezers, and other part will be automated where freezing will be done automatically. Now if you look at this, this is the another unit which is called that glow discharge unit, where you will take your grid and prepare your grid for freezing. So this glow discharge unit we are going to use and make our grid hydrophilic, then we will take this grid here to prepare our sample. Here one thing you can see, this is a unit where we will keep our liquid nitrogen container here, which will go up and help us to prepare the sample. Here this is the two blotting papers are there which is going to blot the sample. Here this is basically a foot paddle. Here we are keeping it up and using as a hand paddle, but most of the places it will be at the bottom and you can use this as a foot paddle. So now I am going to introduce two of my students. They are going to show you how to glow discharge your grid, how to prepare your cryo sample, and then after freezing your sample, how you are going to make your cartridge and put it inside the cassette and transfer it to the microscope. So here is my two students. One, Drosh. Drosh is here in this microscope facility for last 2 years, and he is an EM manager. He is maintaining this cryo-EM facility and preparing sample regular basis. And he is Clayton Fernando. He is a third year PhD student. He is going to fourth year, and today they are going to demonstrate how to prepare cryo-EM grid, glue discharge the grid, and how to prepare cartridge and inside the microscope. So from now, these two students will take care rest of the part.
Hi all. In this part, we are gonna see how the glow discharge is. We are gonna do how to do the glow discharge. Basically, we are doing glow discharge in order to create a hydrophilic nature onto the grid, so that the samples will adhere onto the grid into a much easier way. Initially, we'll see a closer look how to take the grid and how to do the glow discharge. Now we have took a couple of grids for your reference. Here, this is a quantified 1.2 by 1.3, and this is a gold grid. Initially, sir would have explained you what are the types of the grids. Here, basically, whatever we are using is a quantified and a gold. Apart from here, there is also a carbon coated grid which has a 2 nanometer plus 2 nanometer of a carbon layer on the top. If you look at the grid, this is the top portion, and if you flip around, there is a shiny portion. That's the back side of the grid. Usually, while doing the glow discharge, it is necessary to have the front portion, that is the darker portion of the grid. This portion is where your sample is gonna get adhered, and this part is which we are gonna turn it into hydrophilic using a glow discharge. Here what you see is the glow discharge machine. Basically the pressure, the vacuum, whatever has to be created will be done from this motor, and this is a top chamber where you'll have the grid inside, and this is the operational unit of it. Now we'll have a closer look at the operational unit. What are the settings we have to operate before starting and doing a glow discharge? If you look at the control system of it, there is an initial option to initiate the process and what type of gas we are using, and the topmost is the current pressure value, and the bottom value is what is the desired pressure it has to reach, and what is the current, what is the milliampere we are utilizing for the entire glow discharge process, and what is the glow discharge time, and what is the hold time before the glow discharge has to be started. Initially, we are gonna do this glow discharge for 90 seconds. So initially, we are going to the program menu and clicking next. Here you will get the value for the process time and the hold time. We are gonna change the process time to 90 seconds. So click on it, clear the existing value, go ahead with the 90 seconds and click enter. So once the desired time limit has been set, click on auto to set back to the home screen. So now we are ready to initiate the glow discharge. Now we are gonna keep the grids into the glow discharge machine. Basically you have to take this top chamber out. We are gonna take the grids, whatever we took out. We are keeping it into the glow discharge machine. Once sealed, you can initiate the process by clicking auto run. Here basically whatever you are seeing is the plasma charge which is happening during the glow discharge. This plasma charge will create the hydrophilic nature onto the grid so that the samples will get adhered onto the surface of the grid.
Today we are going to see negative staining. The class highlights today about how the sample will be prepared for the negative staining, followed by the insertion of sample onto the microscope, then screening and the data acquisition part we are going to see step by step. Let's get into the video. So the glow discharge grids we loaded onto the tweezer. Once the tweezer is loaded, we have to take the diluted protein, whatever we purified, or any other biological sample. Then 4 microliter of the sample will be loaded onto the top surface of the grid like this way. Once it is loaded, 1% urinary acetate we have to put in the parafilm like that way. One more time. So in such a way 3 droplets we can take. That's it. Once we apply the sample, we have to wait approximately 30 seconds for the sample to settle onto the grid. Then we have to take this grid and then we have to blot out the excess buffer and all like this way. Once it is blotted out, we have to swirl onto the urinary elastic like this. The same pattern, it has to be repeated twice or thrice based on how strong the staining you need. The last set you can do more time without touching the surface of the bottom. Once it is done, again you have to remove the excess staining. Then you have to keep it in room temperature for evaporating the excess staining.
Once the grid is dried, we'll load the grid onto the sample holder. We'll unclip this. And load the grid. We'll place it onto the holder and adjust it such that it is in the center, and then clip it back and verify if it is fixed properly or not.
Okay.
For inserting the grid, we have to keep this pin into 11 o'clock position and insert it halfway inside. The light starts glowing. This position is called parking position. Now we wait for the vacuum to attain. Once the vacuum is attained, the light stops glowing. So now we will insert the holder into the column. For that, we will rotate this anti-clockwise such that this pin comes to the 6 o'clock position and we'll insert it. So this way, the holder goes into the column. Once the insertion is complete, we will switch to the systems. Here we'll acknowledge single tilt and then open the column valve to see the beam. We'll insert the screen, and once we see the beam, we'll bring it to the center and adjust Z height using wobbler. Once the Z height is set, we'll go for the data collection, which we'll be doing in the next session.
In this session, we have seen the anatomy of the cryo-electron microscope, which is nothing but Talos Attica. Followed by we seen how to do the glow discharge to make the grids hydrophilic. Then we seen how we will prepare the sample for negative staining. Then followed by we seen the insertion and overall view of how we will collect the data acquisition. In the next session, we will see how we will prepare the sample for cryo-EM, which is nothing but plunge freezing. Thank you.